Our next speaker is Professor Sanjeev Singh. Sanjeev Kumar. He is professor at Department of Biotechnology, Soviet Pansila University, Haryana. He has been recipient of many awards, especially from NIH. I see CV full of awards by NIH for the wonderful work he has been doing. Professor Sanjeev Kumar.
Good morning and salutation to one and all present here. You know, many times I just write my name Sanjeev Kumar and my wife Professor Gupta is writing Lanka Gupta. So many times people call me Sanjeev Gupta. Yes. Today, somehow the gentleman give me Sanjeev Singh. Okay. So I'm really thankful to the organizers because they give us a chance to visit their university after a longer time and meeting the great personalities in science to discuss the work. So I'm going to summarize the work what we have done with the drawing some conclusions. So we know that we are born in the world of microbes. They are present everywhere. They can survive in any situation or condition we cannot even think of. So in this situation, how we have to survive? So that was a point we thought of to find out the reasons and how this is possible. And one fact we have to know that we are all born sterile. That before birth we don't have any microbes in the body, but as soon as we are out in this world, the microbes are encountering. Some of them are beneficial, some of them may not be. As you can see this picture with the age, the microbes is increasing, and this is a data about the gut of a human being. So many of the bacteria or microbes, they enter through different pathways of the body and they house inside or outside. And we believe that we have to survive with them because this is the world meant for microbes, not for us. They came first, we came later. So these are the different routes by which they can enter our body, and many of them are called endogenous because they survive here. And they perform many functions, and one surprising note is that they are, especially if we talk about the gut, they are 10 times higher in number to the eukaryotic cell we carry in the body. So having such a huge load of microbes, how really our immune system manages? And getting the benefits from the microbes and not getting any harm. So it is a kind of very fine balance which is required. And especially when we talk about the gut, you know, if this fine balance is not maintained, we end up in some chronic. So this self-tolerance, what I talk about, means not harming to the cell, rather harming to those microbes which are pathogenic or harmful to us. And in parallel, not killing those microbes which are beneficial to us. So let's think about it. It is a kind of very complex question itself, and how the system manages it. Because if we end up not managing this path or this mechanism, then we end up in some kind of inflammatory bowel diseases. That means the immune system is start reacting with the microbes which are present in the body. And it is a devastating situation for someone's life. Because we cannot survive with the microbes. And if we start reacting with the microbes, then there is a real problem starts. So we try to understand that this situation is named as inflammation energy. That means the immune system is there in the gut, but it is not under the process of activation. And the activation of the immune system is also called inflammation. So energy means inactiveness or non-reactivity. So we try to find out the question of such type of questions in mosquito system, because I worked at NIH as well as in present laboratory I am working on mosquitoes, especially on some peroxidases in the gut and how they handle the situation with bacteria and plasmodium. So first of all, we thought to compare human and mosquito, and we found the mosquito and human both are similar in terms of housing the microbes. As you can see, in human, at different parts of the body, microbes are present, And in mosquito also, they are present on the body, they are present in the respiratory system, they are present inside the gut and so on. So we thought it can be a good model for us. And on the other hand, we all know that mosquito, they take blood. So first of all, we analyze that when they take blood, what happens to the microbes that are present in the gut. And we found with time, there is an increase in the number of microbes. So after blood feeding, the mosquito's gut was dissected at different time points and the bacterial level was determined by using 16S rRNA levels. Now, in parallel, we found that there is a specialized peroxidase gene, and this is a secreted peroxidase, and this gene was also induced along with the time kinetics of blood feeding. So arrow, this triangle shows you that when this gene expression was very high, the level of bacteria was low. But when the gene expression was coming down, and around the 24th hour, you see the bacterial level was high. So what we thought after seeing this result, that somehow this peroxidase is acting like a myeloperoxidase or eosinophil peroxidase because they are known to be antibacterial. So with this question, we moved ahead to identify the role of this gene, and we thought to silence this gene. And the silencing was achieved by the double-stranded RNA-mediated RNA interference, and then we analyzed its effect on the bacterial growth or bacterial level in the mosquito gut. So we take 24 mosquitoes, one they were controls and one they were silenced. And we look for bacteria present in their gut, and what we found that the level of bacteria was suppressed. But we were expecting that there should be an enhancement. Then we took the individual 24 mosquitoes and analyzed each individual gut, that how much microbial load they have. And now again we see this result here, we say that the microbial level is going down after silencing this gene. So that was a big surprise for us. That means this gene is somehow not only silencing we can achieve. So you can see we have a pretty good silencing, about 80%. Then in those samples we look for some of the genes which are called immune genes. And you see all the immune genes are upregulated after silencing the HPH15, which is a gene peroxidase I have already discussed. Now we thought to give antibiotics to the mosquito and doing the same experiment. And again you see in the first left hand side, blue color is giving you control and the pink color is giving us the results of the silence. And what we see again, the silencing is pretty good, and there was no increase of these genes, neither in control nor in silence. That means the microbes were not present in the gut, that's why these genes were not induced. So we did many experiments, but because of the shortage of time, I came with a model, and what we found that this is the picture of gut, and gut epithelium is mentioned on the top. So gut epithelium is immunoreactive. As we have already said that this is an organ for digestion as well as for immunity. So making a balance between the two phenomena, digestion or immunity, is very important for every organism. Because the main purpose should be the digestion, not the immunity. So what we see here that gut epithelium, what cells, what they do, they secrete mucins. And this mucin is cross-linked by tyrosine to tyrosine residues of one protein, another protein, tyrosine residue, and it makes dityrosine. And that dityrosine make a network over the epithelium. So directly the antigens present, the microbes present in the gut lumen, which are increased by blood feeding, they are not coming in direct connection or contact with the epithelium. So when there is no recognition, then there is no reactivity. But when we silence this gene, we found that this dityrosine network was broken. It was not developed at all. That means now the antigens are in directly connection or directly contact with the epithelium, and if they are recognized, then immunity is induced. So this barrier concept was really well taken, well accepted by the scientific community, and this modulation, how it is really impact all organisms. So I try to explain that also here, that how we have to survive in the world of microbes. The one thing is that immune system keep tackling with them, killing and removal. So while killing and removal, we produce many toxins like superoxide anions, nitrates, and so on. So they may be harmful for the beneficial bacteria as well as for the host, because we know chronic inflammation is caused by such kind of molecules. Or there may be another way that we have to make a cross-linked barrier between the immune system and the microbes. So they do not see each other, they do not react with each other, and they live happily. It is a kind of policy, live and let live. So get the benefit from them, do not kill them. So this work was very well accepted in the scientific society, as I said before. Now, a report came from Imperial College London after 5-6 years of this work, and what they found that this peroxidase enzyme is also helping the sperms or male gametes in the mosquito. So before I come to this conclusion, I would like to show you here that how the reproductive system works in mosquito. As you can see that this is a female mosquito, and these are the ovaries. They have a common underneath and to the outside. What happens is mating happens in these insects once in a lifetime, and the sperms are stored in a specialized organ that is called sperm. Now, when this mosquito takes the blood, eggs are matured and they passes through the arrow which has been shown by the blue color. Now, the sperms are passing through the arrow by the red color, and at this time, or at this point, these eggs are fertilized by the sperms and they come out of the body and they develop a new life. So I again repeat that only once in life there is mating, and these sperms are stored for weeks. So this group which I talk about, what they have found, that mating, no mating, or bread feeding, how this gene is really induced or regulated especially in the spermata, where sperms are stored. So let's compare this structure with the gut. In gut we say microbes are there, gut epithelium is there, which is immunoreactive. Here again, spermatica is also has a covering of the cells, and the sperms are stored in that lumen of that covering. So no mating on the first lane, you see that this gene was not induced, but when there was a mating, this gene was induced. And when there was a blood feeding after mating, this gene was highly induced. That means this is somehow playing a role with the viability of the sperms. Now what they have done it, this is not my work, this is their work. What they have done it, that in 2 experiments, they look for the viability of the eggs that were shed by the females. So only it is a given fact that those eggs which are fertilized, they survive, but those eggs which are not fertilized, they do not survive. Because in the eggs there is a specialized phenomena happen, they undergo melanization after they are released from the body. In other words, when they come out of the body, they are white in color, but after half an hour, they have become black. That is all because of the melanization. So those which are not fertilized, they do not undergo melanization, so they do not survive in the nature. Now, what you see here, there are 2 experiments in control and silence. They silence this gene in spermatheca and look for the survival of the eggs. And what they found, that infertile eggs, as you can see on the left hand side, the percentage of infertile egg was higher in sinus mosquito. In 2 experiments they have shown it, and there was a statistically significant difference. So now again I thought about, because they have not discussed these things in very extensive, they discussed my paper very well, or rather our paper, Dr. Lentha was also with me in that paper. They have mentioned, but they could not find that what was the reason that this was protected to the sperms, the spermatica. Now I found a picture from some sources, and you see this is the structure of spermatica, and this is the outer covering, which has many cells including the epithelial cells. And what you see in this reservoir, there are male gametes present. Now cuticle is present here, and I found in some other references that this cuticle is having the same property like it can undergo the cross-link. So I believe this cross-linking is responsible for again making a barrier and inhibiting the infection of male gametes with the immunoreactive epithelium. So such kind of a compartment within our body, they are making, probably, this is my assumption, by making such kind of barriers, an immune zero environment. And this immune zero environment is responsible for the growth of microbes for the benefit of the organisms. And with these words, I really thanks to the organization one more time, all the audiences which are listening here, and funding agencies, British Pilani, because I wrote before at British Pilani, and Carolina Barlas Muli, and along with her we wrote at NIH, and Madam Lalita, she is sitting here. One of our students, he has been enrolled here, and Google, scientific media, and all platforms where from I learned. Thank you very much.
Thank you so much. Now the
This might be me being ignorant. I work with fruit flies, and that's where I think it's coming from. So in fruit flies, often after mating or after the kind of nutrition you provide them, there is a gut growth that we see. The size of the gut basically increases. So the change in the microbiota load, do you think is it related with, or first question, are there any changes in the size of the gut upon blood feeding or upon mating in mosquitoes, and whether the increase in the microbiota content that you see, is it related to that increase in the gut growth as well or not?
But we have found that mosquito can take almost 6 times larger blood volume through its own weight.
Right.
So extension, that's why so engorged. So after just allow a mosquito to feed on the skin, and you see they engorge like anything. And then you hit them. This really plays up on movement because they cannot fly. So gut microbiota really changes after the feeding behavior, because in Drosophila they are never blood feed animals. So they have mostly the fermenting bacteria or rotten food. But in case of mosquito, they have bacteria which can lyse the blood. So hemolytic bacteria are there, which flora changes out. But with mating, gut, I am answerless. Really there is no correlation. I believe because they are both different organs, so probably there is no direct correlation. Sir.
Both must be very different, mosquito or human. Is the mucus layer very similar in both?
We did not find any such a strong mucus layer, but there is a mucus layer which is always formed after the blood is taken. I'll tell you one more thing in addition, because you asked a very specific question. Okay? Like when the blood is taken, there are some pre-made proteins. They are all thrown into the blood, and what they do, they make a matrix around this blood which is called peritrophic matrix. And after that, the mucin is secreted, and that may be crossed into there. So there are 2 barriers in mosquito. In Drosophila, no 2 barriers. Now we have been asked a question here that why there are 2 barriers. So one barrier has a larger pore, the peritrophic matrix, and another barrier has a very small pore that can allow only the digestive enzymes to come to the gut. Not even bacterial initiators are allowed to enter into the gut because their pore size is small. So Drosophila situation is different because they never take blood, they always take the rotten food.
Peritrophic matrix is
By these microbes, and they secrete IgA and other molecules to kill them. That's how they control that bacteria cannot enter into the mucus. So I was wondering whether mosquito also has similar kind of situation as we have.
Like in our
Yeah, and certain there are
cells, yeah, membrane cells. In mosquito are there cells which do not have microvilli, and they are different from the normal cells, and they are similar to M cells. And some reports I read, not my work, and they say that when bacteria is not present, then this mosquito cannot really handle very well with plasmodium. So bacteria again they is required to enhance the immunity in such a way like humans do.
Wolbachia you're talking about?
I'm sorry?
Wolbachia.
Wolbachia, yeah. Wolbachia was introduced to the salivary system because the plasmodium at the last ended in the salivary gland.
Aedes has this system.
I'm sorry?
Aedes.
In Aedes it was done. In Anopheles also.
Anopheles also it was
done.
done.
But it was introduced to the salivary. Not in
So what I have understood, so can I change is a transient change whenever the blood meal is being taken, the whole system changes in order to, so it's like, it's a dynamic change which is happening with feeding and otherwise.
Yes, answer is yes. It is a transient change which has cross-linked barrier in when there is no blood feed. Because generally the mosquito feed on sugar. And in sugar fed, it was hard for us to look for and find such kind of structures. Because the bacterial load is very low, that may be one of the reason. And in addition, we found some of the immune molecules are all the time turned on. So they control the population of the bacteria at that time space. But when there is a blood feeding, the bacterial level is high. And that only happens after this peritrophic matrix formation and the cross-linking happens. Because before that, again, those primarily immune molecules are keep on killing them.
So the number may will be similar in one organism.
No, sir. The composition changes. The microbial composition.
So for changing composition, there has to be influx from somewhere. So I assume that they are there. Their number will change depending on the food, and when the blood is there, they may flourish or they may go down. But the composition will be the same.
Yeah, this work we have done. Dr. Lalka has done this work, and they found there is a huge variation in the species of bacteria after the blood meal. As in a human, we also know the kind of blood, the food we are taking, the gut flora also changes. And that controls our physiology and psychology and many things.
But the change in number, not species.
No, in the species also.
Species
also. There is a
also.
species change infection, there is a species.
But I'm talking about the food.
Normally, food, yes. Sugar
Composition.
yes.
Composition changes, number also changes. Like hemolytic bacteria, their number goes very high after blood. But before they are in tiny number.
So that's what
That's what we are saying. Number will change, but
The composition.
Composition.
But the-
Composition.
Like the sugar degrading bacteria, they are
There is a regional
In terms of
species.
variation, no, new species will not come in.
That's what I said. That's what we said.
No, because
have-
blood is sterile, so new species
That's
are
what.
less likely to.
I have one question, maybe very stupid question.
No, sir. Questions are
Like I missed.
Sanjeev.
You have worked with the female, as I see. When you said blood meal, when they're taking the blood meal,
Female mosquito.
female mosquito. But what happens in the male? Male never feeds on the blood. Do they have microbiota
Yes,
in
they
the gut? And how does that dynamics of the microbiota change in the male?
Because males are generally neglected in mosquito.
That I agree, across species.
Yeah. So it's a mind, but this is true that female and male, they have almost similar kind of microbes when they are sugar fed, because they generally take nectar from the and in laboratory we give them sucrose. So general microbiota is similar, but some of the specialized microbiota are present which are hemolytic in a low amount, but their amount increases as I told you.
The question was, when you give them sugar food, does the dynamics of the bacterial load changes in the male also, or you haven't looked at it? Like in case of flies, we don't work with males because they are completely intractable. We attempted, but we cannot track them at all. So we gave up. as usual with all males of all species, you cannot track them. You can track them.
No, that is true that in the female it is very well reported that there is a change in microbiota after the food is changed. And in males generally if you keep restricting them in the laboratory and another thing is they do not survive for a longer time. They die in 2 or 3 days, female survives for a longer time. So less likely there is a change because the diet is restricted in laboratory as well as outside they take nectar. If they change the nectar from one flower to another flower, then they might encounter different kind of microbes. This is my question.
One last question is with respect to association of microbes with higher organisms, insects and maybe vertebrates and so on. Do you think this association between microbes and higher organisms has evolved just once in the evolutionary timescale, or it has evolved independently across different genera, different species over time?
You know, human is the most notorious animal born on this earth. I am saying because I am human. Because the kind of physiology and the food behavior we have changed, our physiology has not been changed accordingly. You take example बहुत गंवार वाला example है. गाय को लीजिए 10,000 साल पहले भी घास खाती थी, आज भी घास खाती है. लेकिन इंसान ने अपने आप को कितना change किया. How much we have changed our food behavior. You can see last 20 or 25 years nobody knows what is pizza, what is pasta, what is macaroni. But now it's not only those foods, no other food. Roti chapati तुम पहले खाया.
But sir, they still eat chapati and
But not
Objection. That's a serious objection.
No, you have grown up. I have an example of my son. Give him pizza all day, 1 month pizza. No, but my mean to say is that the food behavior is changing, and that's why the microwave food is also changing. And you know, and then again on the top, different diseases they also have a change in the microwave flora and the gut and other parts of the body.
Thank you so much, sir. Yeah, you have one last
Do you
question.
perform RNA knockout in larval stages or in adult stages? So what would be the difference if it had been performed in larval stages? Because sometimes maybe at that stage the mosquito acquires bacteria and the bacterial environment is starting to develop. So would there be a difference?
You know, when we talk about metamorphosis, there is a huge change in the gut of the mosquito. And it has been found that mostly the bacteria which are present in the larval stage, they are almost invisible in the next stage. So there is a complete transformation. But it's possible. But now this thing is like how longer this silencing will remain effective. That is another question. Because see again, you two parents, kids are born with the silenced. But in Drosophila and the mosquito, it is not known for that.
Thank you so much, sir.
Thank you very much.
Now I would request Professor Mallikarjun Shakara to felicitate Professor Sanjeev Kumar for delivering such an outstanding.
Okay.
How many times you have been speaker and Dr. Gupta has been chair?